So, thanks to the organizer for this invitation. For me, it's a great honor to be here today, speaking about a cross-border study of the functional consequences of PAX6 mutations, missense mutations. So, since in the early 2000s, our group started to perform the PAX6 analysis, and in 20 years, we reach out a cohort that is formed by monolateral and bilateral aniridia, VAX syndrome, Peters anomaly, morning glory syndrome, microphthalmia, and coloboma. And as you can see down here, this kind of phenotype doesn't have any kind of mutation on PAX6 gene. But monolateral and bilateral aniridia are very representative of mutation of PAX6 genes. There are mutation on choline region, missense, nonsense, frame shift, and also on splice side. The cases of VAX syndrome are positive for deletion of PAX6 and MUT1 gene. And but up today we have the 50% of the cases with aniridia without any molecular diagnosis. So until 2 years ago, we performed this kind of analysis with the Sanger sequencing for permutation and to evaluate the presence of deletion of the chromosomic region of PAX6, we perform an MLPA. So if you focused on the missense mutation and the loss of function mutation that are here represented, this is our chart, you can see that there are only 4 missense mutations, and just one have the threonine 394A. So there is a threonine that turns in alanine, is located in the transactivation domain, the PST domain of the protein. That, as you can see, is not, it's got a different shape, an unstructured shape that is completely different of the pair main domain, the pair domain structure and the homodomain. So we want to investigate this, the potential deleterious effects of this mutation. So we started with the in silico prediction. Through the ACMH variant classification, we can show that this variant is classified as pathogenic. He is located in the exon 12 of PAX6 gene. There's nothing about the scientific literature about this mutation, and it's also located nearby a splice site of the exon 12. So we want to investigate if there is something, if there is an effect on the splice site. We also saw that through different population database, this variant is not reported, and also that the threonine that turns in alanine is in a very conservation domain through different species. So we also performed MUT score for this mutation. MUT score is a pathogenic predictor that interrogates futures trained with a random forest approach. So we are talking about machine learning. And subsequently testing against various databases for hereditary condition. So more the score is near 1, more is the pathogenic effect of this variant. And so as you can see, the score is 0.876, so very pathogenic. This is another landscape, so also always about the MUT score. So this is the position of our missense mutation is nearby 1, and this is the benign. And so as you can see here, is benign, and here is not benign. So here we started also to use another open source predictors. And as you can see, for the missense deleterious, they are scored there. They are very high level of deleterious effect, but we haven't got anything about splice site for this kind of predictors. So we want we performed bioinformatic analysis through the splice AA. That is a 32-layer deep neural network that predicts splicing from a pre-RNA sequence. So as you can see, this works like the mood score. So more likely one, more is a spontaneity score prediction. So as you can see, all the scores are near 0. So it seems like that there is no effect on the splice side. So I will show you the preliminary data of our project. We have the pleasure 6 years ago to meet our colleagues from the Russian team. They work in the research center for medical genetics in Moscow. And during the Duisburg meeting, the third conference, an Iridia conference, we met them and we started talking about this project because they are very specialized in study the splice sites. So we worked on a project that is also being awarded from the Iridia Europe. And so our colleagues perform the splicing activity, and we perform the transcriptional activity. So for the splicing activity, we perform a mini-gene assay. Our colleagues perform a mini-gene assay, and I think that I can skip the methods because it's technical quite known. And we evaluate the transcriptional activity using MCF7 cell line. That is a human breast cancer cell line. And because this kind of cell doesn't express endogenous PAX6, so we want to eliminate this kind of bias. So we also perform the dual rapid assay system just to check with the PAX6 mutated against the PAX6 wild type combined with the element P3 and CD19 of the promoter gene of the receptor to produce activity. So this is our results. For the splicing activity, we saw there is no difference between the control and the mutated as a splice AI prediction. So as you can see, the mutated construct is at the same weight of the wild type one because there is retain of the exon 12. The band was isolated and also sequencing, Sanger sequencing to evaluate the presence of the mutation. So the splice AI prediction was correct. But for the transcriptional assay that we perform, we can show that there is a significant difference between the activity of PAX6 wild type versus the activity of the missense mutation. So there is a decrease of the transcription, and this because PAX6 mutated bear the mutation that this is a confirmation that this mutation is deleterious. But what about the negative cases? The negative cases, as I told you before, we got 50% of cases and then got molecular diagnosis. Yeah. So in the last 2 years, we reached a new technology that is the NGS sequencing, and we started until the last year with a custom panel with 36 genes related to the disease, but now we started to perform clinical exome and for some cases whole exome. So we started to evaluate our cohort, our negative cohort, and now I want to show you a very interesting case about a patient with an iridia, and as we know, clinical exome allows us to evaluate this single nucleotide variation and also the copy number variation. So we saw, as you see in this report, there is a deletion that interests the MAF gene. MAF gene, this is a span of the mutation, and the ACMG CMV classification predictors say us that this is likely pathogenic. We also confirm the presence with a real-time PCR. As you can see, there is the number of the deletion of the patient. And we also performed the segregation analysis in the parents, and we saw that this deletion is de novo. So the health parents haven't got this deletion. What we know about the MAF gene is that the transcription factor, the CMAF, that plays a very important role in the regulation of embryonic lens cell development, with a very high interplay with PAX6. So MAF gene point mutation, we know that we are associated with congenital cataracts, but there is nothing about the deletion. So in vivo models and human phenotype indicate a very heterogeneity of the phenotype, because we could be a microcornea, standalone, iris coloboma standalone, or maybe both, and other segment dysgenesis condition. So for conclusion and future prospect, we saw how in silico prediction and deep learning are relatively new in techniques in biology, and are not without trade-offs. So we always need an in vitro experiment to evaluate the functional, with a functional study, the pathogenicity of the genetic variant. And also we saw how new technology strongly help us to reach new data and so to identify the causative mutation, but this data that turn back to us could be conflicting data. So this kind of data must be evaluated in parallel with a highly detailed clinical evaluation of the patient, of the phenotype of the patient. We need it. So, in conclusion, I want to thank our colleagues for this great collaboration. This is just at the beginning. And I also want to thank NVIDIA Europe and the Italian Association for the support and for the trust in us. And I want to thank you, all of you, for the attention.